Thank you for joining us today. This is the eBioscience Affymetrix CITO 2015 commercial tutorial. Today we are honored to have two guest speakers from the Cincinnati Children's Hospital Medical Center, Sherry Thornton and Monica DeLay. They will be speaking to you about our RNA by flow cytometry assay, PrimeFlow. Our first presentation is from Sherry Thornton. She is an associate professor at the Division of Rheumatology and also the Flow Core Director. She will be presenting on her research and how it has benefited from the integration of the PrimeFlow assay. Our second presentation is by Monica DeLay. Monica is the Flow Core Manager at Cincinnati Children's Hospital. She will be presenting on a number of the different applications of the PrimeFlow assay, along with the tricks and tips to help establish this assay within your core. We hope you enjoy these presentations. Thank you.
Well, I will thank Dylan for inviting us. I feel privileged to be able to present this, and we're really excited about this technology that we've been able to use in our lab. So I'm going to talk a little bit, like Dylan said, about the project that we're using this for in the lab, and then Monica's going to talk about putting it into the core and other specific examples of people that have used it in our core as well. So I just want to talk about, I am not a physician myself, so I do not diagnose people with systemic JIA. But Alexei Gram is the physician that I've been working with for quite a while. He is an expert in diagnosis of systemic juvenile idiopathic arthritis and macrophage activation syndrome. Dr. Schulert, I'll be talking about some of his work as well. He is focused on looking at microRNAs and their effects on macrophages as well and polarization, and we'll get into that in quite detail. Nate Fall is our research assistant, the hands in the lab, and Alyssa Sproles is the technical person who runs the PrimeFlow assays in the flow cytometry core. We are supported by grants from the NIH, the National Institute of Arthritis and Musculoskeletal and Skin Diseases, and our core is supported by the NIH as well. So a little bit of clinical background, just so you can relate some of the things I'm going to show you. The systemic JIA was formerly known as Still's disease. It is considered a subtype of juvenile idiopathic arthritis. It's characterized by daily fevers and associated with arthritis in at least one joint. You also have one or more of these presentations, an effervescent rash, which is shown here, or enlarged spleen or liver, or also lymphadenopathy or serositis. And the onset is early in childhood. It's usually between 1 and 10 years of age, and it has a slight female predominance. It is considered an autoinflammatory disease rather than an autoimmune disease. And it is characterized by innate immune and monocyte dysfunction. So you get increases in IL-1 beta, monocytes, or IL-1 beta, IL-18, and IL-6. You do have activation of monocytes and neutrophils. We also see an increase in the infiltration of T cells and bone marrow as well. So T cells and monocytes are involved. And this is kind of a schematic of what could be happening. We don't know the exact mechanism yet, but we know that CD8s are expanded. You get soluble CD8 increase and soluble IL-2 receptor alpha. You also get cytokines that can activate macrophages, including especially interferon gamma. And these macrophages become highly activated, and you can get secretion of soluble CD163. You can also get this kind of cytokine storm where you have TNF alpha, IL-6, IL-1, and IL-18, all of those are upregulated. Now when this really goes awry, you get a symptom known as macrophage activation syndrome. And that syndrome can be fatal in the kids that get this. It can be fatal in up to 25% of those kids. So we're really interested in what is going on with these macrophages. And these are hemophagocytic macrophages, which happen in the macrophage activation syndrome. They can engulf white cells and nucleated red blood cells. And these hemophagocytic macrophages are CD163 positive. CD163 is a scavenger receptor. It recognizes hemoglobin and haptoglobin complexes. And here it's shown on the surface of the cell. This is just a schematic of the macrophage. And it can bind to these hemoglobin haptoglobin complexes, release iron after endocytosis. This iron is sequestered with ferritin. And the reason why I'm showing you this is, first of all, the CD163, but also in macrophage activation syndrome, you see high levels of ferritin that is sequestered with iron. That's one of the clinical parameters as well. So we're interested in macrophages, and classically what has been, I guess they've been characterized as either M1 or M2. So M1 are involved, it's classical activation, and you get TNF alpha and IL-1 beta produced, and you get pathogen clearance. And then M2 is another alternative activation where you get resolution and repair in these kind of macrophages. So we're wondering what kind is actually in the systemic juvenile idiopathic arthritis in the disease. So the new view, however, of the M2 is that there seems to be several types of alternatively activated macrophages. And these are just shown here. You have M2A, which is the classical alternative activation, and that's involved in wound repair. The M2B, which are involved in immunoregulation and IL-10 production, and M2C, which produce IL-10 and TGF-beta. So what do we know about the monocytes and systemic JIA right now? We know that they actually have markers of both phenotypes, the M1 and the M2. So we're very intrigued to figure out, can we associate these with active disease and clinically inactive disease? So for the M1 markers, we've seen IL-1, IL-6, TNF, and CD40, CD80. And then you also see markers of alternatively activated populations. Okay. So we're focusing on this surface marker CD163 because we see it in these hemophagocytic macrophages in MAS. And we're wondering, or we're trying to figure out how this molecule is also regulated. So we've been looking at microRNAs, and there has been recent work that has identified microRNA as an essential regulator of macrophage polarization. These are, I'm sure everyone's heard of microRNAs, but they are small non-coding RNAs. They're about 22 nucleotides in length. They can bind to the 3' untranslated region and target it for degradation. As high as 50 to 90% of genes may have some microRNA regulation. So we know that the microRNA can regulate macrophage differentiation. So we actually looked at systemic JIA monocytes in both an active versus inactive disease, and we looked at 50 specific microRNAs that are involved in innate immunity. We found that miR-125A5P was one that was significantly elevated in children that had active systemic JIA as compared to clinically inactive disease, and this is shown here. If you look at the clinically inactive, which is the CID patients, and this is their level of microRNA, you can see that they have much lower levels than active systemic JIA. And the NOS here is new onset systemic JIA. So both of these are highly active, and you can see that this MIR is highly upregulated in these monocytes. This also correlated well with the clinical parameters, including arthritis and rash and hepatosplenomegaly. So what is miR-125A5P? It's evolutionarily conserved. It is a microRNA cluster. It has been implicated to be increased in M2 polarizing conditions, and it's essential for mouse polarization to M2. So we took, this slide is a little busy, but we took both an antagomer, which will suppress miR-125A5P expression in the monocytes, and then the next slide that I'm going to show is overexpression of miR-125 to see what would happen if we took a cell line, THP1s, and exposed them to different conditions, whether they were M1, M2B, or M2C. And so we're looking at M1, and you can see when you add the antagomer, which is the gray bars, that we don't really see any differences here. And with the open bars are untreated, the closed bars are a negative control antagomer. And then we have, so that would be how these different markers are upregulated during those M1 conditions. And so we don't see differences here. We do see differences in M2B polarization. We see CCL1 expression that is decreased when you actually decrease the integument. And then when you overexpress the integument, you actually see that M2B is increased. So we also see a difference in both CCL1 and CD163. We see CD163 increased in the M2B conditions and in the M1 conditions actually. And we see it downregulated in the M2C conditions. So what this tells us is this mirror or microRNA seems to be a controller of macrophage differentiation depending on what milieu it is in. So we then want to figure out, okay, with the systemic J, what's there, what's triggering this, what kind of phenotype can they get then. So we want to look at CD163, okay? And I think I've hit that over the head for you guys. But we do want to look at CD163. So we've looked again in THP1 cells. These are the different treatments that we've used. And we found that IL-10 for 24 hours increases the expression of CD163 by real-time PCR. And this is real-time PCR data, as was the previous 2 slides. We then took a clone of CD163 and looked at it by flow cytometry, and we only saw a slight shift. So we were, okay, what can we do to look at this in a different way? And then in comes the prime flow. So we decided to look at the prime flow for CD163. They have a nice probe for this. So I'm going to go through the technique, basically, of the prime flow. And the prime flow, it's a unique integration of the cell surface markers that you've been doing all along and being able to bring in analysis of gene expression. So then you can look at these in tandem. You can look at up to three different RNAs by a standard flow cytometer with your cell surface markers as well. You can also do intracellular antibody staining as well, and Monica will be showing you some examples of that. It is different from the SmartFlare technology. The RNA in the SmartFlare can be detected in live cells. This, you immediately fix the cells and you have RNase inhibitors that inhibit the degradation of the RNA. Okay, but with the SmartFlare, you also have to be able to endocytose that probe and take it up and let it sit overnight. So how does the signal actually come about? Because that's been the limiting factor. You get a sample preparation, you primerize the cells, you unmask your RNA, then you have your target that you're specific to. Here they're shown 2 RNAs. Then you have probes that come in and bind. The probes are usually to about a kb of sequence in your gene. And you have a 50-mer here that are 2 25 oligomers that will bind together. And then you'll get branching, a branching technology which allows the signal to be amplified up to about 8,000 times. Okay, and then your floors will come in, and this is your 1 RNA floor, and then you add your other RNA floor, and these are the available probes as of now. You have Alexaflor 647, which is a type 1 probe. Type 4 is Alexaflor 488. And Alexaflor 750 is the type 6. And what we've been using is Alexaflor 647. So again, just real quick, the assay, the workflow for this, you're gonna prepare your samples. You can label with your antibodies before you fix the cells, or you can label after. It's up to you, and you gotta work out those kind of conditions. Then you bring in your target, and you incubate your cells with the gene-specific probe. You have a signal amplifier here, and then you're going to detect it with your flow cytometer. It does take a lot of spins. That's one thing, and I'm sure Monica's going to talk about this a little more. But this is our data. So I want you to concentrate on the right-hand side first. For every probe that they give you, or that comes with the prime flow kit, you get a control probe, which is shown here with our THP1 cells, and most of the cells took up that control probe really well, or hybridized that control probe really well. Here we have unstimulated and stimulated, so again we stimulated with IL-10, and you can see here that in the unstimulated cells we still have CD163 RNA that's present in the cells, but when you stimulate with the IL-10, you get a nice threefold increase, which is similar to what we saw with the real-time PCR. We also looked at this with ImageStream, and here is this, and the green cells are the CD163 positive cells, or the cells that were treated had the probe, the CD163, and the blue are ones that did not have the probe, and these are the positive cells. Here are the negative cells that we did not see the A647 in. So you can see the bright field and then the AF647, and you don't see a signal. And here you can see the nice signal from the AF647 probe, which when you include the bright field, you can see that it's internal to the cell. So like everybody says, seeing is believing. So it is there. So what are our future goals? We have used CD14 packed blue. I didn't show the data here. But that works well with the AF647 CD163 as well. We want to use more cell surface markers, and we're in the process of doing that right now. Eventually, we want to look at peripheral blood mononuclear cells of systemic JA and MAS and see if there's any differences in those, in the expression of CD163, CD14, CD206, CD209, and other macrophage differentiation markers. The other thing that we want to do, since we've seen the microRNA regulation of CD163 by real-time PCR, we also want to confirm that by flow, which I think will be a nice
So I'm going to talk about some other applications, some of them from examples from Affymetrix, some of them from our institution. We've really only just begun. So we're starting to get adoption of this technology from users of our core, and we're starting to try to expand that a little bit. So I'll kind of talk about some of the things we've been seeing, but also an example, which I don't have any data from, but of something that we're going to be working on in the future. So, okay. So we just saw an example of cell phenotyping along with gene expression, which is really, really powerful because you can now drill down into a single cell level, but look at the kind of the heterogeneity of the phenotypes along with the gene expression. So there are some other applications that I just want to give some examples of the ways that you can circumvent antibody limitations by using prime flow. Looking at the kinetics of RNA and protein in the same cell, and also the detection of viral RNA. And then I'm going to show some examples of some other work from our core. Okay. As you can imagine, certain species don't lend themselves very well to antibodies that are commercially available. So this is an example of someone that's interested in catfish, and they're looking at viral infection in catfish with relation to the aquaculture industry and pathogens that might be prevalent in that industry. So this person, she's from University of Mississippi, and she has one antibody that is available to mark NK-like cells in the catfish. And the top row are the CC41 negative cells. The bottom row are the CC41 positive cells. And you can see that she had a number of probes made that were available to her. So she was able to really look at these NK-like cells and look at specific probes in those cells. And you can see that some of these are positive as you compare them to the CC41 negatives. So this is one example. And also in our institution, we have a developmental biology department that does not use traditional animal models that we'll be working with in the future as well. One of the really elegant ways that I feel like you can use prime flow for is looking at transcription and translation at the same time in a single cell. So here is an example of CD8 positive T cells from human PBMCs. And these were stimulated with PMA and ionomycin and harvested at different time points. What they looked at was interferon gamma and TNF alpha, mRNA on the y-axis and protein on the x-axis. And you can really start to look at the kinetics of how transcription and translation occur in these cells. For example, at 1 hour, the interferon gamma mRNA has increased, but there is really no protein expression quite yet. While at 2 hours, you're starting to see both mRNA and protein come up. And then you begin to see decay of the mRNA at 3 hours, 4 hours, and 5 hours. So then if you compare that to the TNF alpha expression, you can see that very rapidly by 1 hour you can see both mRNA and protein expression, but it looks like the mRNA begins to decay at an earlier time point than the interferon gamma. So you're beginning to see that at 2 hours, and then quite a bit, there's almost very little transcription going on here, but you still see some that are double positive, but you're beginning to see more that are just positive for the protein. So I think this is a really powerful way to use the technology. The other thing that I just want to show, because this is an interesting study, there's a group from Stanford that's interested in hepatitis C, and they're looking at a wild type strain and a mutant strain. And they were able to develop probes that were specific for each strain. And you can see that when they infect hepatocytes with the wild type strain, they're able to detect with the probe. I think this is type 1, so this is the Alexa 647 probe. In the mutant strain here, you can see the cells that are infected with just the mutant strain. This is the type 6 probe, which is the Alexa 750 probe. When they mix cells together that have been separately infected, they can detect both of them. And then when they co-infect hepatocytes, you can see that they are now double positive. So they're positive for both of the probes, and they're able to detect that. And this is correlated nicely with the microscopy image. So you can see the type 1 lighting up in the cells that were infected with the wild type, the type 2 lighting up, or type 6, which is the mutant strain, lighting up here. And then you can see the merged images. Now to one of our investigators. We have Dr. Claire Chenier, who's in our immunobiology department, and she studies Tregs with respect to HIV infection. And one of the things that, they're interested in looking at some transcripts via flow so that they can phenotype their cells along with this. But they wanted to start off with an RNA that they were familiar with, so looking at FOXP3 expression. So just sort of a proof of principle to make sure that they could get the assay to work. So here we're looking at FOXP3 mRNA on the y-axis and protein on the x. And what they did was they took from frozen stock a human CD4 positive enriched PBMCs, and they stimulated these cells overnight with CD3, CD28 beads and IL-2 to induce the FOXP3 expression. So if you look at, and these are unstimulated, if you look at the unstimulated cells, and this is just the mRNA probe only, you can see that there's a little bit of basal expression of FOXP3. But in the stimulated cells, and what they were particularly interested in doing is, I'm sure a lot of people know that FOXP3 is an intracellular protein. And you have to, a lot of times when you purchase the antibody, it comes with a specific buffer to use to stain the cells. So they were interested to see which worked better, if they used the FOXP3 specific buffer for staining their cells before going through the prime flow assay, or just using the prime flow staining buffer before going through the assay. And so that's what the comparison is here. So this sample is showing protein staining with the prime flow buffer, and then adding the mRNA probe. And then this is using the FOXP3 specific buffer and then going through the prime flow and staining for the probe as well. So you can see that these are virtually identical, where you have quite a bit of RNA expression in these cells after stimulation and a little bit of protein expression for the FOXP3. So this was a great way for them to get started, and they're going to be working on some additional things. As I said, I don't have data for this, and I wish I did. One of our investigators, this is a really nice way to bridge a lot of different technologies, so I'll kind of try to tell you the story. The investigator that is doing this, they're particularly interested in a bone marrow population, granulocyte macrophage progenitors, and the relation to blood cancers. And so in our facility, they sorted the GMP population and took them through to the Fluidigm application and did single-cell RNA-seq. And they found quite a few transcripts that they were interested in and would like to move forward and use the prime flow to phenotype the cells, along with looking at the detection of these particular transcripts that they're interested in. So I think this is a really nice way to complement single-cell RNA-seq data and validate that with the prime flow assay. So they've started to do some of this, but we've been still working out the panels. And so I don't have really any data to show you yet, but I think it's an excellent way to use the technology. So that's something that we're going to be working on in the future. So let me just go over some of the critical assay parameters that one needs to keep in mind when performing this assay. It's a pretty straightforward assay. It's a 2-day process. There are a lot of steps, a lot of spins, a lot of washes, but there are definitely some things that you absolutely have to follow. And the protocol is really good at keeping you aware of these things, but temperature stability is very important. So the annealing temperature for the probes is at 40 degrees Celsius, and this needs to be very steady temperature. So the incubator that you use has to be validated using a temperature validation kit to ensure that the temperature does not fluctuate more than a degree. And so they have a suggestion for a temperature validation kit that you can use. But essentially, and we've been using a hybridization oven in our facility, but just making sure that not only does it not vary over time, but also there's no hot or cold points within the incubator. The other thing that helps is if you have a heat block, and that is suggested as well. And that'll kind of insulate your tube so that when you're going in and out of the incubator, that the temperature is not fluctuating very much. Floor choice is also important. As I said, with the stem cell example, we've had to kind of play around with this panel. So that's why I don't have some wonderful data to show you. But the other thing to keep in mind, most floors are compatible, but you cannot use per-CP or per-CP tandems, as well as QDOTs. They don't survive the fixation method. So that's another thing to keep in mind. Also, antibody compatibility. So depending on if you're staining your cells first or fixing and then staining your cells, you just may want to keep in mind. Actually, eBio has a really nice chart on their website for different antibody clones and which fixation methods they are compatible with. So if you're going to be fixing first and then staining, that's something, or sorry, staining and then fixing and then staining, that's something you can use as a resource. Controls, as we all know, in flow cytometry are very important. They have developed a compensation kit, so they're eComp 1 beads. They have taken that compensation kit and then added the probes that they use in the prime flow assay so that you can just bind those to the beads as well. So you don't have to use your cells, you don't have to use the positive control probe or your probe and waste those reagents. And then those compensation controls are spectrally matched to the probes that you use in the assay. So it's the Alexa 647 the Alexa 488, and the Alexa 750. The other thing is FMOs, especially fluorescence minus probe, so I would call it FMP. But you want to make sure that, especially if you have a lot of different antibodies in the panel, you want to add all those antibodies, take that sample through, but don't add the probe to that sample. So that can provide an excellent gating control for your analysis. The other thing to keep in mind is, as I said, this is a 2-day process with lots of spins and washes. You can imagine that you're going to lose a lot of cells during this process. So the best way to minimize that is to use a swinging bucket rotor. We actually did a comparison between the swinging bucket and a fixed bucket. Definitely do not use a fixed bucket rotor. That's all I have to say. So swinging bucket rotor is essential to make sure that you're minimizing how many cells you're losing. The other thing I just want everybody to keep in mind is that during the fixation process, and because of the temperature that the cells are being incubated at, there's an increase in autofluorescence in certain channels, and this seems to be cell type dependent. And we discovered this with the cell type that we were using, but let me just show you first. Looking at human PBMCs and cells that are either fixed only in the black histogram or fixed and then gone through the prime flow process. You can see that a couple of the channels off of the blue laser, you see an increase in autofluorescence, and then all of the channels off the violet had a slight increase in autofluorescence. So again, something to keep in mind when building your panel. But we had the misfortune of choosing a macrophage cell line, and what we found with this THP1 cell line was that we had autofluorescence increase in pretty much every channel. Well, it was in every channel. So there was just something about the cell line that, that essentially did not like the prime flow assay as far as autofluorescence goes. So again, something to be aware of when you're designing your panels or choosing the cell type that you're going to be using. So I don't know if you all have a core facility, but we run a core facility, and our utmost concern is our clients and making sure they're using our facility to its fullest. And so when we see new technologies like this come along, we get excited and thinking how they can use it because we think it's valuable. And it'll add something to their research. So people were kind of afraid to try it because it's a lot of cost in the upfront to try this technology out. So what we decided to do, we had enough people that were interested, and we were able to purchase a kit and then just charge them per sample just to recover the cost. So we didn't make any money, we just kind of a pass-through charge. And that was a really good way to do that. Technical support is really key. It was important for us that we knew how to do the assay, so that's why we took one of our staff members and had them trained to go through the assay. Again, it's not very difficult, but it's still nice to have somebody around that has already done it, and you're not just leaving your investigator to themselves. The other thing I will say is that Dylan, as well as Sue Reynolds from the company, has been great for helping us troubleshoot and work out panel design. Education is always important, so we had some seminars and you know, you could do a workshop or hands-on training. We've offered for people to follow us through in the lab if they're interested. And marketing, because it's not so much we want to sell something, it's that we think it's beneficial to our users. But education is a form of marketing. It's actually a very valuable way to do it. But we do some email blasts to let people know that we have this reagent available in our core, and if people are interested in trying it out, it's available for them to do so. And then we have tried to make sure that we're highlighting data that's generated in our core. So it's not just a, hey, look, this company has some data and we want to share it with you. It's this PI at our institution is using it, and this is how they're doing it. So in summary, it really is not a difficult assay. It's easily adapted by users. It provides a unique niche for the core. As I said, we can kind of branch out into those departments that traditionally have not used flow cytometry because there's no antibodies available for their model organisms. But it's also, not the traditional way to use flow cytometry with antibodies. It's looking at RNA. And then, it really allows you to look at gene expression at a single cell level, and I think that's really the power of the assay. So.
Yeah, hi, I'm Katja. I'm working in Dresden at the Center for Regenerative Therapies. And yeah, I'm just heard that nice, lovely talk about the prime flow technology. And I'm working in a core facility, fax facility there. I attended that talk not to necessarily try that technique on my own, but to know what's going on the market and basically help users and know what they are doing, and maybe introducing that new technique to our facility as well. So that was actually a good suggestion, and just buying and trying it ourselves and then just offering the people who come to us, trying it out.
I'm Mike Lee from UCSF. It's a pretty good presentation. I've heard about this technology before, but it is nice seeing how it is applied in a real-world situation, just from not hearing it directly from you guys. But no, I found it pretty useful.
Thank you for watching our presentations today. For more information, visit www.ebioscience.com.